today I'm gonna introduce an ongoing project that involved with Dr. Pingwen Shi to study the glucose homeostasis. And the title for today is Ventral Medial Hypothalamus Glucose Sensing Neurons and the Glycemic Control. I want to use this title page to summarize that. Really, it is the changing of firing activity in the glucose sensing neurons that control the glucose homeostasis. My lab in Louisiana State University focuses on identifying the novel ion channels, neurotransmitters, and neural circuits in the brain that are critical for the feeding behavior and glucose homeostasis. Last time when I was in UIC, I present the project that as person regulate feeding behavior. So this time I want to switch to this glucose homeostasis project. In our project, we combine the transgenic mouse models, electrophysiology, in vivo calcium imaging, chemogenetics, optogenetics, and other cutting-edge techniques to study the neural function in the field of obesity and diabetes. The major technique we use in the lab is electrophysiology. We can do multiple cell types. Although majority of the cells we focus on neurons, however, we also collaborate with other investigators. For example, we record isolated beta cell for Jason Collier in our institution, and we also help Dr. Shi and Dr. Jiang in UIC to record the fat cells currents. There's another small structure we can even do the recording on the sperm cells. So overall, we just want to fully use our electrophysiology technique to help others. Now, I want to start our story for today with this in-flight hypoglycemia cases. So there was one passenger who traveled alone. He got an emergency on the flight with symptoms of being pale, slurred speech, and dizziness. Luckily, there was onboard doctor who found out that was because the overdose of insulin from his insulin pump. Luckily, he was rescued by then. But this case reminds us that in some cases, the hypoglycemia could be very dangerous, especially in those type 1 diabetes patients. And we need more effective ways to prevent the severe hypoglycemia. So that's when we start to go interested in this research. And our normal subject has a very good mechanism called counterregulation, which can fight against the significant change of blood glucose drop. So it's called counterregulation. When the hypoglycemia happens, a group of neurons in our brain can sense the tiny change of glucose drop. They will conduct the signaling to the downstream organs. On one hand, pancreas alpha cell will release more glucagon, and on the other hand, they will release less insulin. Another organ they will target is adrenal gland. They will release more counterregulatory hormone, including cortisol and epinephrine. Those counterregulatory hormones will target to liver to increase the glucose output and target to muscle and fat to reduce the glucose utilization. And the most studied neural brain region I showed here is called the ventral medial of hypothalamus. It was reported that a lot of glucose sensing neurons are concentrated in this small area. So I just want to give you an impression where this brain region located if you are not familiar with the animal research. And there's multiple papers already demonstrated that the glucose sensing neuron in ventral medial hypothalamus are important for the counterregulatory response. Take this diabetes paper published in 1995 as one example. What they did is they directly implant a catheter to target the VMH region, and then they injected 2-deoxyglucose to induce the glucopenia. Upon injection of 100 millimolar 2DG in VMH, they observed that there's a huge increase of blood glucose. Well, compared to the normal glucose, like 5 millimolar to 10 millimolar, there's no change of the glucose. And more importantly, they observed some counter-regulatory hormones increased after the injection of 2DG in the VMH. So including the epinephrine, norepinephrine, and glucagon. So this data really indicated that the neurons in the VMH are important for the counter-regulation. In our hand, we published a paper in Nature Medical Communications to identify that another group of neurons that specifically express the estrogen receptor alpha. You will observe that those neurons are highly concentrated in the subdivision of the VMH. We call them ventral lateral part of VMH or VLVMH. And interestingly, we discovered that all of those neurons are responsible to the glucose. Either they are identified as the glucose inhibitor neurons or GI neurons, or glucose excited neurons or GE neurons. So we identified there are around 60% of neurons express ER alpha in VLVMH are GE neurons, while only 40% are GI neurons. And those neurons will work together to more regulate the glucose homeostasis when hypoglycemia happens. And there are other people also reported different neuropopulation in this small area VMH. They are functionally regulated glucose homeostasis. For example, some people published that PACAP expressing neurons, they are glucose-sensitive neurons, including GE and GI neurons. And as fun neurons in VMH are also important for the glucose homeostasis. Recently, Martin Meyer's group published a paper in JCI to indicate that the CCKB receptor expressing neurons are glucose-sensitive neurons. They are able to regulate glucose homeostasis. But overall, all those discoveries involve the glucose-sensitive neurons only represent a certain percentage of the neuron, including GE or GI. Our previous study just identified they are mixed with part of the GE and part of GI belongs to glucose-sensitive neurons. So we come up a question, can we locate or identify a novel neural population? They have even higher, ideally 100%, but maybe not, to be exclusively like GE or GI. And in this case, it will purely respond to the glucose changes. And the answer is yes. Recently, we identified a group of neurons that express this vascular GABA receptor. We call them VGAT neurons. They have very high percentage of neuron identified as GI, like over 87%. So that's the beginning of this project. To give you an impression where those neurons are located, we have a reporter mice that cross VGAT pre with TT tomato. You can observe that most of the GABA neurons are located on the ventral lateral part of this VMH. It's a very unique region that they concentrate these neurons in a small area. And this is consistent with the database, the Allen Brain database. But before we studied this neural population, nobody noticed and people trying to ignore it because most of the neurons in the VMH are identified as the glutamatergic. So there's no discovery about the GABA neurons there. And so to directly verify if those neurons respond to glucose, we use our electrophysiology method. We can directly record the single GABA neuron in the VLVH by targeting those red fluorescent expressing neurons to do the whole cell patch clamp. And what we discovered is they also mixed with two neural populations, including GE or GI. Showing here is two representative potential neuron traces from GE or GI. You can observe that the GI neurons are activated by the low glucose switch from 5 millimolar, which considered as the normal glucose level in animals, to a low level of 1 millimolar. And the Wolff spikes shown here represent the action potential. The more spikes you see here means the neuron have higher firing activity. So what we observe is in the GI neurons, the neurons are activated when we switch from 5 millimolar glucose to 1 millimolar glucose. And in the GE neurons, they are inhibited with reduced firing frequency of action potential. And I think this is an important discovery because we observed that a large percent of those GABA neurons are GI neurons, which is very different from the previous publications. Only a small portion, like 3.8, they are identified as the GE neurons. So we believe that unique distribution of the GABA neurons may have functional relevance to the glucose homeostasis. And as control, I give you some other expression. For example, the GABA neuron in the other brain regions, they are not like the region in the VMH. They mixed with GE or GI and almost like half and half. Also, other neural population, for example, SF1 neuron in VMH, they are half GE and half GI neurons. So there's come up another question with our previous discovery. Do those GABAergic neurons overlap with the ER alpha neurons because they are all located in the small area? And in order to answer this question, we collaborate with Dr. UIC here. And what he did is he stained the GABA-A receptor by using their ER alpha Z-screen mouse model. So you can observe that there's only limited neuron population. They have both red signal and green signal, which means there's limited neural population. They express both GABA-A receptor and ER alpha receptor. And in our hand, we also got TD tomato reporter mice. We record the neural activity in response to a ER alpha receptor agonist called PBT. By doing this, we are able to identify whether this group of neurons responds to the agonist of ER alpha. So we identify whether they are ER alpha receptor expressing neuron or not. And our data showing that is very limited neurons response to PBT. So that means large percentage of those GABA neurons, they don't express ER alpha. So they are independent two neuron populations. Thus, in this project, we hypothesize that the GABA neuron represent a unique population of glucose inhibiting neurons, which are activated during the hypoglycemia, and its function critically contribute to the counter-regulation hormones, counter-regulation response. So to directly record those neural activity under physiological condition, we introduced this hypothalamus technique. What we did in this experiment is we got pre-animals, inject a pre-dependent AAV virus, contain the GCAM6 fluorescent vector. And in this case, only the pre-positive GABA neurons will express the reporter of this GCAM sensor. Once the free calcium binding to the GCAM sensor is gonna increase the density of the fluorescence. So we can use the photophotometry system to measure their neural activity correlated with this density change. Shown here is a validation of the injection site, which target to the ventrolateral part of the VMH. Now, in the physiological experiment, we give the animal two injections. One is the steady injection, which serves as control. You will see this black curve. They don't change the neural activity in the GABA neurons. However, when we give the animal with insulin injection, which can induce the hypoglycemia in mice, and we saw the GABA neuron increase the neuron activity, which means when animals go through the hypoglycemia, this will trigger the activation of those GABA neurons. But the next big question is how blood glucose changes when we manipulate the firing activity of those GABA neurons. This will give us some more answers whether this population of the neuron are important for the glucose homeostasis. So in the neuroscience field, there are two popular manipulation of the neural activity. One is called optogenetics, the other is chemogenetics. I think they are using a pre-dependent vectors so they can manually activate the neurons either by light or by chemical, for example, CNO. Now in our studies, I only want to give one example of the channel-rhodopsin, which is an activation type of the protein that sends the blue light only. Once blue light stimulate this protein, they are going to trigger the potassium influx, and then it will activate the neurons. On the other hand, the chemogenetics will involve two different types of the receptor. One is called HM3DQ, which is the activation type of the receptor. The other is the inhibition jet called HM4DI. But I'm not gonna give a detailed explanation. We're gonna use the experiment to go through this. So in this experiment, we are using the light-sensitive, pre-dependent channel-rhodopsin vectors. We injected those vectors in the ventrolateral part of the VMH, and showing here is a validation image that we can selectively express only in the ventrolateral part. Once the animal recovered from the surgery, we will deliver the light through this optical fiber, so neurons can be manipulated whether triggered to activate or no change. So what we did here in this experiment is we deliver a pulse of 10 milliseconds blue light to stimulate those neurons for only 50 minutes. And during this time period, we measure the blood glucose every 5 minutes. We observed that once the neurons are activated by the blue light, it has significantly increased the blood glucose compared to the sham light yellow, because channel-rhodopsin does not respond to the yellow light. And even we stop the stimulation, this trigger of the increased blood glucose can last for more than half an hour. More importantly, we set up another group of mice to do the repeat experiment. At this time, we just stimulate animals, and then we quickly using that animal collected their serums. We detected that when this group of neurons are activated by the blue light, it has increased the glucagon but reduced the insulin without change of the corticosterone. And this data indicate that activation of this group of neurons will trigger the counter-regulation hormones. And next, we do another experiment by using the chemogenet. What we did here is we selectively express the inhibitory jet receptor in the ventrolateral part of the GABA neuron. Shown here is a validation image to validate the expression level of this inhibitory jet. Now, different from the optogenetics experiment, this time we introduced the 2DG induced the glucopioneer experiment. But because it takes time for the CNO to inhibit the jet expressing GABA neurons, so before we give the animal TDG, we inject the CNO 30 minutes ahead. And then what we observed here is in the control animal with normal function of those GABA neurons, 2DG will trigger an increase of blood glucose in mice because it's gonna induce the glucopioneer. However, when GABA neurons in the VMH are inhibited by the jet, the animal cannot increase as much as the control animals, which means the GABA neuron in VMH contributes to the counter-regulation response. So I think by using these two type of data, we can conclude that the GABA neuron in VMH is important for the counter-regulation. Now, the next question will be, do those GABA neurons also regulate feeding behavior? Because think about it, when your blood glucose goes down, on one hand, you try to increase the blood glucose. On the other hand, you try to seek for food. And then it also triggers the increase of blood glucose. That's most of the time happen side by side. Well, unfortunately, we did not observe a significant feeding behavior change when we stimulate or inhibit those group of neurons by using jet. And it's some sort of disappointing. However, one explanation is the function of this group majority focus on the glucose homeostasis, but not the feeding behavior change. So that's our current conclusion. But We may also seek for further regulations. For example, we will challenge animal by fasting and then repeat this experiment. Hopefully, we get some other discoveries. Now, to summarize the first part of our research in this project, first of all, we identified a normal neural population in the ventrolateral part of the VMH. They are inhibited by high glucose and activated by low glucose. Secondly, hypoglycemia can significantly activate those neurons in vivo. That depends on our photometry experiment. And when we manipulate those group of neurons, and we will find that activation of those group of neurons will increase the blood glucose, while blocking them will impair the 2DG-induced counterregulation. And of course, we don't observe any feeding behavior changes when we manipulate those group of neural activities. Now, the next big question is, what is the intracellular mechanism of hypoglycemia that activate those GI GABA neurons? That's also another interesting question most people ask me when we present our research. So, well, we still come back to use our electrophysiology because that's very quick and directly can answer this type of question. In this experiment, what we did is we screened multiple different ion channels, and lastly, we identified probably the calcium channel would be one of the intracellular mechanisms. So we designed experiment here showing in this slides. We first treat the GABA neurons with normal bath solution contain 2.4 millimolar group calcium ions. And we observed that most GABA neurons are GI neurons because low glucose activate their neural firing activity. However, when we switch the bath solution with zero calcium, which means they don't have the free calcium in the solution, so animal cannot, the neuron cannot use the calcium to trigger the downstream signal pathway. And interestingly, under zero calcium solution, low glucose no longer trigger the change of the firing activity. It means this change requires the calcium ions in the neurons. And when we switch back, the normal bath solution contain normal calcium concentration, we can restore this increase of firing activity by switch the glucose from 5 to 1. So overall, this experiment indicates that the calcium signaling is important for the glucose, especially GI neurons, to detect the low glucose change and activate neural activity. Now, we wanted to specifically ask the question, what type of calcium channel are involved? We screened some sort of other type of the calcium, and we believe this T-type of the calcium channel may be the target because they can respond fast. And what we observed in this experiment is when GI neurons are treated with low glucose for 1 millimolar glucose, it will activate the T-type calcium channel current amplitude only in GI neurons, but not in GE neurons. And if we block the GI neurons by incubation the CAP3.2, which is a subtype of the T-type calcium channel blocker, then we failed to see the changes of this firing activity caused by 1 millimolar glucose. And this data indicated that maybe the subtype of this CAP3.2 would be the target for low glucose to activate the GI neuron activity. However, someone would argue that the pharmacological blockers are not so selective to support your conclusion, and we have to use a better method. Now, in the next experiment, we introduced the Cas9 CRISPR technique. So what we did in this experiment is we get Cree mice and injected the both sides of VLMH with AAV Cree-dependent Cas9 guide RNA, which targets the CAP3.2, followed by a reporter of mTRI. In this case, only the selective pre-positive neurons in ventral lateral part will be deleted for the CAP3.2. As control, we use scrambled guide RNA, which followed by a GFP reporter. So we are able to use electrophysiology to perform the recording. And our data shown here is in the knockout animal, in the control animals, we still observe a large percent of the GABA neuron are activated by low glucose, which identified as GI, which is consistent with the previous data. However, in the knockout neurons, this population decreased from 84% to only 14%. And largely of the other neurons are non-response to the glucose change. And you will see that the GI neuron population percentage does not change at all. So this data conclude that when we knock out CAP3.2, it's only changed the GI neuron glucose sensing properties, while not affect the GE neuron population. And more importantly, as I mentioned previously, the GI neurons can be activated by low glucose, but inhibited by high glucose. So we did an opposite experiment. It's just by treating those GABA neurons with high glucose. Then in this case, ideally, GI neurons can be inhibited when we switch the glucose from 5 to 10. Now what we observed is in the control neurons, it was inhibited when high glucose treated. However, when those neurons have CAP3.2 knocked down, 10 millimolar glucose no longer inhibits their neural activity. So this data majority argue that CAP3.2 are critical for the GI neurons to sense the glucose change and change their firing activity. And showing in this figure B, when we knock out CAP3.2 from the GE neurons, which although they are small population, however, they don't change the glucose sensing properties in those GE neurons. Now, overall, this electrophysiology data conclude that CAP3.2 ion channel will be an important regulator for low glucose to change the GI neuron firing activity. Now, you may ask, what is the physiological function role of CAP3.2 in the GABA neurons? That's our majority purpose, because most of the time we want to introduce our basic discoveries from the in vitro to the in vivo. And in this experiment, we generate another group of animals by injecting the control vectors or the guide RNA target CAP3.2. What we observed is after 2 weeks of virus injection, we start to monitor their food intake every 4 days and their body weight. Well, interestingly, we don't observe a significant change of the food intake or body weight when we remove this CAP3.2 from the GABA neurons. This data, somewhat consistent with our previous finding when we use JET to activate or inhibit those neurons. They do not really change the feeding behavior. What about the glucose homeostasis or counterregulation? Then in this experiment, we generate a new batch of animals, including control and knockout mice. We give them IP injection of 2DG to induce the glucopenia. And we found that in the control animals, they have good counterregulation response. So animals have increased the blood glucose. However, when the GABA neurons have deletion of CAP3.2, 2DG will only induce lower level of glucose change compared to their control animals. This data means that CAP3.2, number one, CAP3.2 are important for the counterregulation. And number two, this looks like it's not the only role. There may be other existing intracellular mechanism because we don't see a complete abolish of the counterregulation response. All right, to summarize the second part of this result, we identify a subtype of the voltage-gated calcium channel called CAP3.2. They are important for the glucose sensing properties in the GI neurons only, but not in GE neurons. And deletion of this CAP3.2 from the GABA neurons impairs the glucose sensing capacities. And more importantly, selectively deletion of CAP3.2 from the GABA neuron does not change animal food intake and body weight, which is consistent from the previous GI experiment we observed.
I'm sorry, can I ask you one question? Sorry to interrupt.
Sure.
When you do the 2DG experiment, is that injected peripherally or centrally into the brain?
That's a good question. So here in this experiment, we inject peripherally.
Peripherally, okay.
Yeah, so it's IP injection.
Yeah, no, I think that's really important because that's supporting the idea that you're sensing differences in glucose availability peripherally, not just when you overwhelm the neuron locally.
That's also great. Some, we considered some other reference published, but that's a good question.
Yeah, thank you.
And so lastly, selectively deletion this type of a calcium channel subunit will significantly impair the counterregulation. As I mentioned, this may not be the only mechanism for the GI to regulate because we are not able to completely abolish the counterregulation in mice. Now, let's come back to our previous mention about this hypoglycemia emergency case. So our long-term purpose is to identify an effective method to prevent the severe hypoglycemia. And from our current discoveries, we believe this small group of the GABA neurons could be one potential target for the prevention of severe hypoglycemia. However, whether how and which method we can use will still need more time to develop. And of course, we can consider to target only the CAP3.2 ion channel because this ion channel can, if you have an ideal chemical which inhibits or activates this channel, it may induce less side effects. Now, for the future plan in this project, we are still working on to identify or narrow down a more selective gene marker for these GI neurons. As I mentioned previously, what people discovered in this field is most of the neural population identified to be the glucose sensing neurons, they are mixed with GE or GI. Our current project only significantly identified a neural population with very high, but not ideal. Ideally is whether we can look for a neural population, they are 100% GI. Maybe we cannot identify a neural population, but who knows? So we try our best to do the single cell RNA sequencing because this type of experiment will give us more details about the gene markers within those GI neurons versus the GE neurons. We have collected sufficient samples after the electrophysiology and send them to the genetic code for the single cell RNA sequencing. We now have some data indicate one certain of the gene, which I cannot share with you today, but hopefully when we publish the manuscript, we can share with you. They have very high percentage of expressed in the GI neurons, and we believe that will be an even better marker for those GI neurons. But just let you know, we are working on this, and hopefully we have a better gene markers to represent the GI neurons in ventrolateral part of VMH. And second question we want to follow up in this project is, what are the downstream neural circuits of those GI neurons in VMH? And we have a very good method called the CRACM, or the channel-rhodopsin assisted circuit mapping, to dissect the large complex neural circuits with optogenetics. So we have the plan to selectively express the AAV-dependent channel-rhodopsin mixed with a transnepsin marker called WGA or ADINWED. The beauty of this method is, first, you will have the pre-positive neuron in the ventrolateral part of the image express both chondrodopsin and the WGA. The chondrodopsin will not only express in the cell body, but also the fiber terminal. On the other hand, the WGA will transnapt and express the fluorescent GFP to the downstream neurons. So if we look for the identify downstream circuits, we just need to screen the whole brain and look for the neuron burn region, which have high density of the green fiber terminal. That will be the neuron regions that receive projection from the via of image. And of course, we have this WGA positive neurons, and then we can use electrophysiology to identify whether they receive the monosynaptic projection from the GABA neurons. I think we have generated a lot of data. Unfortunately, because of the time limitation, I am going to show you next time if I have chance. But we are following up this, and this project will also collaborate with Dr. Xu here, and he did a lot of circuits mapping and also physiology right now in his lab. So lastly, I would like to acknowledge my close collaborator, Dr. Xu in UIC, and of course, other collaborators currently like Dr. Jiang and Dr. Liu. So we have multiple ongoing grant proposal, which focus on the diabetes and peripheral tissues, which hopefully we send out for to send out the NIH 1 grant proposals. So most of the work was done by my postdoc, Dr. Feng, and research associate Emma. We have 2 undergraduate students who performed the feeding behavior monitoring. So I appreciate their help. Mandy just moved from Pennington to University of Florida with her husband, and we are currently hiring more people. Lastly, I would like to acknowledge the funding source from NIH. Without the funding from NIH, we are not able to generate those data I presented, and hopefully we will keep getting more funding from NIH. Now, with that all, I'm happy to answer your questions. Thank you.
Maybe you could repeat it in case it might not be able to hear it. So first question is, how do you think glucagon speeds?
I have a question. Am I missing one live there that I don't hear, or I don't want to interrupt?
Maybe let Terry go first. So Terry, can you repeat your question?
Yeah, I will ask a question. If I interrupted someone, I just want to mention that I couldn't hear them. And so if they ask a question, could you please repeat it so that we people who aren't there also can hear their questions? Okay. Yeah. So the question I had was, it relates to some of the work Mike Schwartz presented to us a while ago. And he's really focusing on fasting glucose levels in the brain as a glucose sensor, and which tries to stabilize fasting glucose. And, you know, his work is focusing on mechanisms involved in sensing fasting glucose levels, and in models of obesity, you know, he thinks the set point is changed, and in part due to changes in glial reactions